Is it possible to observe organ development in the lab, model diseases, and find the right drug for the right patient? Traditional approaches, such as 2D cell cultures, animal models, and imaging techniques, have moved science far ahead, but they have several limitations. For example, 2D cell cultures lack the cellular diversity and three-dimensional architecture of human organs. The physiology of model organisms like mice is different from that of humans, which can lead to erroneous conclusions about the efficacy of drugs. And human imaging techniques face challenges related to resolution and artifacts. Recognition of these limitations has spurred a hunt for a more physiologically relevant model to conduct research. That model is organoids. Let's take a closer look at them. An organoid is a three-dimensional assembly of cells created in a dish that can self-organize and replicate some aspects of organ structure and function. The creation process begins by isolating somatic cells from a patient. Reprogramming makes it possible to generate from them so-called induced pluripotent stem cells, capable of turning into any cell type in the body. Next, these cells are grown in colonies. In a commonly used approach, the cells are then separated from each other and placed into round-bottom wells to allow them to sink and aggregate. This is how spheres called embryoid bodies are formed. Replenishing the media with the right cocktail of nutrients and growth factors is needed to specify the desired type of tissue, such as brain, lung, or gut. In the next step, early-stage organoids are embedded in a substance called matrigel, which acts as a biological scaffold. It creates a three-dimensional environment for cell growth and differentiation. With a specific set of growth factors, organoids continue to develop into pea-sized models of real organs, only in simplified form. But how can such models be used? Human organoids have shown huge potential to study organ development, model hereditary and acquired diseases, as well as develop patient-specific drug screening. As a result, organoids address the limitations of traditional model systems, offering a middle ground between 2D in vitro cell cultures and animal models. Organoids are safe and ethical for several reasons. This entire research process is subject to rigorous ethical regulations and informed consent processes. The possibility of transforming mature body cells into pluripotent stem cells avoids the need to use cells derived from embryos. Moreover, organoids can serve as a reasonable alternative to using animals in doing research. Organoids mimic human organ features. They are already being used to study human development, model diseases, and for drug testing. In addition, organoids can be used and researched ethically and present alternatives to animal models. The journey has just begun.